Hello and good day, everyone. I'm Ryan Joseph Felicidario. Alongside with me is my co-author and my thesis advisor, Dr. Ramon Santos, and we are going to present our study, Measurement of Ascorbic Acid Samples Optical Rotation via Improvised Polarimeter for Purity Assessment. The presentation will have the following structure. We'll start off with an introduction of the problem we seek to solve and the concept related to our study. Then, we will discuss the methodology taken divided into four parts: the construction of the polarimeter, the experiment proper using polarimetry and melting point determination, and lastly, the post-experiment analysis. Next, we are going to show and discuss the results in the polarimetry and melting point determination experiments. Finally, we will quickly summarize and conclude our study. Emerging problems associated with drug use and abuse include adulterating drugs and different chemical substances. Drug adulteration is constantly and commonly seen where there is a decline in the purity of most drugs. To detect these adulterants, chromatographic methods are commonly used as they have a high separation capacity to detect multi-drug components in a sampled substance. However, chromatographic methods require special equipment which can be costly to acquire and maintain. Aside from the common chemical analysis techniques available, polarimetry can be used to analyze inorganic and organic compounds. However, Breton 2015 observed that students struggle to understand the concepts due to the black box system of commercial polarimeters. With this, we characterize ascorbic acid to assess its purity using an improvised polarimeter. So what is a polarimeter? With this, we discuss a few concepts. Let's start with light polarization. From high school physics, we know that EM wave consists of oscillating electric and magnetic fields perpendicular to the direction of propagation. When light is directed through a polarizer, electric field components align in a particular direction. The electric field of the light exiting the sheet comprises solely the elements aligned parallel to the polarizing direction. One application of polarization is found in organic chemistry through optical activity. Optical activity refers to a unique property exhibited by chiral compounds like ascorbic acid, where they have the ability to rotate plane polarized light. Polarimetry is used to quantify the extent of this rotation. The tool employed for this measurement is called polarimeter. The mechanism of polarimetry goes like this. First, we have an unpolarized light source. As it passes through a polarizer, it becomes polarized. This polarized light will be rotated once it hits a sample containing chiral compounds. Finally, the angle of rotation will be measured using an analyzer. This optical rotation is usually expressed in the standard way called specific rotation, denoted by square bracketed alpha. Specific rotation is defined as the observed rotation when light with a 589.6 nanometer wavelength is employed with a sample path length of 1 decimeter and a sample concentration of 1 gram per cubic centimeter. The calculation of a specific rotation is governed by Beer's law, which is simply the observed rotation over the product of path length and concentration. When the observed rotation is expressed in this way, it yields a physical constant unique to a particular optically active compound. Breton 2015 mentioned in his study that undergraduate students struggle to understand this concept. Hence, several homemade polarimeters have been documented for student experiments. The first demonstration typically involves utilizing an overhead projector. Another polarimeter design was by Mehta and Greenbow. Breton 2015 also made his version where he used two PVC pipes. Another polarimeter design was by Bernard and Mentes featuring a 3D printed body. Furthermore, Lisboa and Sotomayor presented a laser polarimeter apparatus. In this study, the polarimeter design developed by Witting and Jusnes was used due to its advantages. First, its resolution of 0.5 degrees. Second, it requires minimal skills for a more durable setup. Third, its affordability renders it suitable for educational settings. The cost of the construction of this polarimeter is around 6,500 pesos, which is significantly inexpensive compared to commercial and manual polarimeters that cost around 644,000 and 71,000 pesos respectively. Proceeding with the four-part methodology, we start with the construction of the polarimeter, which is guided by our main reference, Wittingen and Jusnes 2020. The polarimeter was assembled with the aid of Lego bricks. For the base of the instrument, four 6x16 Lego plates were connected. The fixed polarizing film was sandwiched between two Lego bricks. The LED light source was held using a 2x1 brick. The protractor was supported with 4x2 and 2x2 bricks. And at the back of the polarimeter, we can see the red LED detector. And the part that we modified in this setup is that we added a resistor in series with the LED detector to improve the detection capability of the device despite having a sample with a relatively low specific rotation like ascorbic acid. Upon constructing the polarimeter, we are now ready for polarimetry measurements. We start by preparing our samples first. Two different brands of ascorbic acid named brands X and Y were dissolved in a certain volume of distilled water such that we achieve our desired concentration, which is 0.25 grams per cubic centimeter. Before measuring the optical radiation in a dark room, the instrument was first calibrated. From this, we determined the optical zero. To do this, two detections were done: visual detection and voltage detection. For visual detection, we replaced the red LED detector with a white paper. The range of angles corresponding to the dimmest light intensity were recorded. To ensure precision, a margin of 4.0 degrees was added on either side of these noted angles. For voltage detection, the voltage reading at the midpoint of the visually detected angle range was initially recorded. Then, incremental adjustments of 0.5 degrees were made on both sides while monitoring the voltage displayed on the multimeter. The angle corresponding to the minimum light transmittance was identified as the optical zero. After calibration, the sample was poured into the glass cuvette with a known path length. To locate the new optical zero, the same method used for calibration was repeated. Then, we get the observation by subtracting the calibrated optical zero from the new one. Note that we added the visual detection method, which was introduced to serve as the baseline for voltage detection. To supplement the polarimetry characterization, the melting points of the samples were determined. This is done using the melting point apparatus. To start off, we prepare the sample by transferring a small amount of the substance onto watch glass. Then, we dried the sample by either oven drying it or pressing it onto a piece of filter paper. Then, the dried sample was introduced into the capillary tube. After that, we placed the capillary tube to the sample holder, as shown in Figure 12. To operate the apparatus, a target temperature of at least 10 degrees Celsius below the expected melting point of the sample and a gradual ramping rate of 1 degree Celsius per minute were set. Finally, the melting range was recorded. This is simply the temperatures at which the samples began melting and completed melting. To analyze the observation for the 5 different path lengths used, the specific rotations were computed. This is done using the equation below. Then for the analysis, the calculated specific rotations were compared to literature values. Note that ascorbic acid has a specific rotation of 20.5 to 21.5 degrees. On the other hand, from the melting range as recorded from 3 trials, the average melting point was calculated using the minimum and maximum values of the melting temperature range. Then, this was compared to the theoretical melting values. Note that ascorbic acid has a melting range of 190 to 194 degrees Celsius at which it also decomposes. If the values are close to the theoretical ones, we will conclude that the sample will likely be pure, otherwise it is impure. Now we proceed with the results of our experimentation. Table 1 presents the calculated specific rotation values derived from the observation. Note that from the literature, the specific rotation is derived from the observation when a light source with a wavelength of 589.6 nanometers is used. However, the peak emission wavelength used in the experiment is 592 nanometers. Therefore, the values obtained are expected only to be close to the literature values. From the measurements, for band X, the percentage error is relatively low, averaging 4.76%. Conversely, for band Y, the percentage errors range from 23.8% to 52.4%, with an average error value of 41.2%. Here's a scatter plot of the observation versus path length for bands X and Y. The plot of band Y deviates noticeably, which can be attributed to some impurities. Note that the slope represents the specific rotation divided by the concentration. For band X, the slope is 5.23, resulting in specific rotation of 20.9 degrees, which is within the range of the theoretical specific rotation. Meanwhile, for band Y, the slope is given as 3.50, corresponding to a specific rotation of 14.0 degrees, which is outside the expected specific rotations. Therefore, band X is suggested to be pure, while band Y is impure based on this polarimetry experiment. In melting point determination experiment, we see that the average melting point of band X falls within the theoretical value. Furthermore, the observed melting range is narrow. Conversely, the average melting point of band Y significantly deviates from the theoretical, and the observed melting range is broad. This suggests that band X is pure, while band Y is impure, which is both consistent with our result in the polarimetry experiment. In summary, this research used an improvised polarimeter to measure the optical rotation of ascorbic acid in order to assess their purity. This primary characterization method was supplemented by melting point determination. Here are the summary of the results for polarimetry. Here's for the melting point experiment. And in conclusion, band X is likely to be pure, while band Y is likely to be impure based on the two characterizations done. This comprehensive approach combining polarimetry and melting point determination proved to be cost-effective compared to other devices. This offers a practical and accessible solution for purity evaluation. Furthermore, the non-black box nature of the polarimeter facilitates a straightforward demonstration of optical activity and polarization concepts. Here are the references we use in this presentation, and that's all, and thank you very much for listening.